We can now move into our next panel. So we're at panel 3.2. This is brain cell atlases for neuroscience research. So we have an hour and a half to focus on two kind of central themes, technical challenges in integrating multimodal data, and also conceptual challenges in using multimodal data for discovery and generating hypotheses. So the panel here will be moderated by Dr. Jeremiah Cohen and Dr. Xiaowei Zhuang. Dr. Cohen is a principal scientist at the Allen Institute and an adjunct. He has also an adjunct appointment at Johns Hopkins. His research focuses on using cell type specific tools and behavioral tasks in mice to probe circuit function. And Dr. Zhuang is at Harvard University, where she's a Howard Hughes Medical Institute investigator, the David B. Arnold Jr. Professor of Science, Professor of Chemistry and Chemical Biology, and Professor of Physics. And her research is focused on developing and applying novel imaging tools with single molecule sensitivity, molecular scale resolution, and dynamic imaging capability to allow direct visualization of molecular interactions in cells, as well as tools that can image large numbers of genes. So I will turn the floor over to our moderators for this last panel. Thank you.
Okay, great. Thanks. I think in the interest of time, we'll keep the introduction brief. But this panel is really about how we use and develop new brain cell atlases for doing neuroscience research. And so this, is really, a lot of this is about multimodal experiments and comparing different properties of cells across experiments to each other. And so you heard a little bit about this from Carl's preceding keynote. And so Xiaowei and I have divided the larger topic into two general themes. One is really about the technical challenges that we all encounter when integrating multimodal data. And as you'll see, a lot of this has to do with space. And as Carl mentioned a few minutes ago, really requires precise and accurate spatial registration as a means of doing this. And then the second theme is about the conceptual challenges that we are bound to encounter when comparing data from multiple modalities. For example, transcriptomic data to morphological data of cells to activity in the context of animals that are behaving, and how we think about those frontiers. So there will be 7 excellent talks. The timing here is a bit draconian, so apologies to everybody, but we want to squeeze it in and leave enough time for general discussion. So we'll try to moderate without having to bring out the vaudeville hook. So maybe I can, unless Xiaowei has anything she wants to add, I can pass it off to Yongsoo for the first talk.
Yeah, let's go to the speakers.
Okay, great.
Since we have a little time, let me just get to it. First speakers, the Karel's keynote presentation nicely highlight the importance of our the spatial framework, and the topic that I choose to address in this session is how will brain common coordinate and anatomic ontology nomenclature be established and standardized to facilitate multimodal data visualization and integrations. And I see broadly 3 challenges here. The first challenge is conflicting anatomical ontology and nomenclatures. And the second challenge is longitudinally aligned CCF across development time point. And the last challenge is how to align CCF across species. So I will provide some of the data and from my lab and some thought how to address these issues. First challenge to address is different anatomical labels from the same species from the same time. As you know, there are multiple different atlas exist for mouse brain, for example, on adult mouse brain. How do you mitigate those? And I wouldn't say one atlas is better than the other. Each different anatomical atlas and nomenclatures utilize different set of criteria and standards. Hence, they also serve their own merit in their own way. But there is a need to bring these 2 different or more than different atlas framework into a consistent spatial framework to compare and contrast. Here, this is our previous works importing Paxino's anatomical labels into the Allen Common Coordinate Framework template. So by importing anatomical label from Paxino's anatomical drawing into the Allen template, we were able to compare the background color is Allen CCF labels, while the white line delineate Paxino's drawing, and how these two different set of the labels delineate the structures differently. But yet they are all in the same spatial context. So as long as data is registered to this one spatial framework, anatomical labels in this spatial framework can be used to interpret those results using different set of anatomical labels. And also to facilitate a multimodal data, registering data from the different modality is quite challenging. For example, light sheet data to the CO2 photon-based anatomical template is rather challenging. Hence, really the next round of Atlas development should consider multimodal imaging modalities as well. And also, it should not come from the single subject. It's better to be coming from the multiple animals with a population averaged from both male and females. In our work, we use more than 10 animals to generate population average during the across 8 different developmental time point using both MRI and the light sheet. And light sheet was co-registered to the MRI. Hence, both MRI data set as well as light sheet data can be registered to this kind of spatial framework. By doing so, we were able to leverage different contrast features distinctly available in each individual imaging modality, such as here, MRI has a lot of different white matter tracks and water molecule informations, and light sheet microscope can provide a lot of detailed cell type information. And all this information can be merged together in the same spatial framework. And moving forward, all this spatial framework need to be in the physical space in the Cartesian coordinate, so it can be also used for the surgical preparation to really understand how the morphology change across the development. And another challenge here is to use ontologically consistent label across development. By doing so, here we extended Allen CCF V3 labels into the early postnatal time point as early as P4. Because this label is co-registered and down all the way to the P4, we can use this ontologically consistent anatomical label to interpret how signals in the developing brain change across time. And the last one is, this is in a sense, grand challenge. How are we going to integrate Atlas framework across different development? And one of the idea is utilizing pilotypic stage. And though different species has a different brain shapes, if you look at early time point, their brain shapes looks quite similar. And here is mouse MRI brains, E15.5, and this is a 13 weeks old human fetal brain. And you can see the morphological similarity as well. Utilizing this morphological similarity, we might be able to reconcile cross-species atlas differences. So key consideration in the CCF, we have to use a standardized template that consider both the sex and individual difference, and multi-contrast template to support different imaging modality. And we need a unified atlas with a different anatomical label in the same anatomical template. And lastly, common ontology and label across species using a developmental shape convergence will really facilitate cross-species integrations. I will stop there.
Great. We can maybe take 1 or 2 minutes for questions for Yongsoo and then move on to the next talk. Carl.
I have a couple of comments. The multi-contrast registration is really important, and having some kind of automated, robust workflows is very important as well for the community, right? I mean, most people don't know how to do this. And of course, the value of the coordinate framework and then linking to all the atlases is if everybody can use it in their studies. And I think figuring out how to collect the best of class templates for registration and maybe also make some good workflows available to the community might really advance this kind of work, linking all kinds of things to the transcriptomic work. That's just one comment. Again, it's sort of a need. The other thing is, I think the registration is incredibly important. The ontologies, to me, a little bit secondary. That will change over time anyways, right? And can be version controlled and all this kind of stuff. But I want to hear your kind of thoughts about that, right? And there'll be probably multiple different ontologies and segmentations that really make sense depending on what you're interested in.
Right, absolutely. So let me start with the registration questions, and it is really important. And one challenge that I notice is when people use different kinds of anatomical signal like PV staining or different cell type staining, it's hard to register those data. So there is common coordinate framework, and their contrast features differs in each users. Hence, what we're trying to build, at least there is a template on the autofluorescence. But there simultaneously there is a Nissl-based template and a GAD-based template, accessory cells-based template. By really truly providing multi-contrast template, all synchronized in shapes, that will really make the registration workflow easy. And there are a set of registration tools become available, more user-friendly, so that tech development need to continue. That I agree. And you mentioned about the cross-species ontology. Sorry, I forgot your question. Can you just quickly rephrase one more time?
Yeah, I think the ontologies, they're going to change anyways, and we just have to have many versions and version control and so on.
Yeah, absolutely. And there is so far, when we look at development, Allen common coordinates is based on site architectures, how to apply all the way down to the embryonic stage. Because in the embryonic brain, things are changing and the things are not settled yet. Hence, for looking at true development very early in the embryonic, we have to use a different sets of the ontology. So we use development based on gene-based ontologies. Hence, based on users' need, different labels that based on different ontology can be selected.
Okay, let's do two quick ones and then move on. So one from Partha.
Hi, hey, Karel. Just wanted to say that I just put a paper in the chat. We've been putting it out for several years, but it's finally out. There is this tool set we have that does handle kind of cross-modal signals, with missing tissue, etc., blah, blah, blah. And there is a software package. It's a bit, involved to use, lots of parameters. But yeah, there is this tool set. If you have any interest, if you have data sets, maybe we can have a chat about that. And one comment I want to make is that the registration will only be as good as two things. One is individual variation. You can't get over that. But the other thing is how much local context do you have in cytoarchitectonic terms? I mean, ideally, let's say you knew the transcriptomic signature of every cell in the neighborhood. Then you could probably, really pin down where you are in the brain and learn a mapping between that thing and XYZ. Of course, such data cannot be gathered usually in experiments, but whatever data you can gather for local context, right, and then learning a functional relationship, that's what the registration problem is really about, between that local context, rich context to the XYZ, that's the way to go. And that's an experimental question, can you gather that context? The autofluorescence signal doesn't have a lot of context in it.
Yeah, so I think it's the same answer. I think so. It's just basically autofluorescent with multiple different registered contrasts that also serve as a kind of a different registration target. I think that will mitigate.
And last one is Hongwei.
So just one comment. Great talk. Like for Atlas, we're constantly evolve. We refine the brain structures. For example, like a striatum. We, based on our connectivity, refined into major divisions, smaller domains. That's in mice. But also recent work, you see, like today, even Evan gave the talk in the human, you can also refine it into the different subdivisions. So I think like at this moment, or because now we have everybody is should be on the same page. We start from beginning. We need to think about how to standardize these kind of nomenclatures rather than like before old time we have Swanson, we have Paxinos, but now since we all work together in this big group, maybe like from beginning stage we can think about how to consolidate this kind of newer subdivision nomenclatures from mouse, from the human, from other primates, and also for the connectivity-based parcellation versus spatial transcriptome defined subdivisions. Yeah, that's just my comments.
Excellent comment. Thank you, Hongwei. So I think the key here is label, as you said, will continuously evolve. It can be final or it can be based on different criteria. As long as we keep the space template, as long as all data is registered to that single space, as long as we agree upon that space, currently for adult brain, RNS itself is to really demonstrate part of that. As long as all the data is registered there and the labels are there, we can compare, contrast, and reconcile. So that's a great comment. Thank you.
All right, good.
Right, great. Thanks, Yongsoo. The next talk is Xiaoyin Chen from Allen Institute for Brain Science. Take it away.
Hi, everyone. So my lab works on developing barcoded connectomics techniques to understand wiring rules in the brain. Generally, these techniques, we basically use RNA barcoding and RNA sequencing to read out connectivity with high throughput, low cost, and single cell resolution. And depending on how you do the sequencing, you can, in many versions of this, you can read out transcriptomic information at the same time from the same cells. And so today I want to talk about what where these technologies are today, and what we like unique kind of questions that we can ask. So here is one of these experiments, the Bosiljka experiment, where so we barcoded a lot of cells in the cortex. If you zoom in, you can see that the cortical cells are filled with, the somas are filled with barcodes. And then if we look in the thalamus, for example, you can see a lot of these are barcodes found in the axons. And so in a sequencing experiment, we basically do iterations of sequencing chemistry followed by imaging. So then for each of these dots and the somas, we can read out the whole barcode sequence by many rounds of imaging. And then we can match the barcodes in the axons to the barcodes in the somas. And that gives us, basically tell us which of these axons are linked to which soma. And essentially, this allows us to reconstruct single neurons in a relatively densely labeled sample. So here are two example neurons, just to give you a sense of the data we get. In each example is a single neuron, and the red dot is where the soma is, and all these other dots are the barcodes found in the somas and the axons. So here you can see, for example, this neuron has a little local dendrites, and then it sends an axon down here, makes a turn here, and then branch out over here in the thalamus. This is IT neuron, so it has an extensive local dendrites and axons, sends an axon down, and then you can see on the other side, you can see the axon in the corpus callosum coming to the other side, and you have this contralateral homotopy projection. So I'm showing you only two examples, but in an experiment, we get thousands of neurons in a single brain, in a single cortical area. And that density allows us to look at the topography in projection, which I think is really important, because I think projections, like topography, is a fundamental organization of projections. It's not point to point. So here we're looking at the same type of data. This is a cell with all the axons represented in dots. So these are the barcodes in the axons. And then I'm just going to overlay more neurons onto this flat map. And if we overlay all the cells, then you get this really nice color pattern. And the colors here represent the medial lateral locations of the somas within V1. So this reflects retinotopy. So this means we can actually look at the retinotopy in the higher visual areas, the projection from V1 to higher visual areas. And if we compare these to functional mappings previously, we were basically able to capture the same patterns. So we're able to essentially capture the retinotopy of projections at a single cell level. So this is all looking at just the projections, but I mentioned briefly that with in situ sequencing, we can actually get gene expression out of the same cells. So then we can look at gene projection relationship at a single cell resolution. So just to show you some examples, here we're plotting a bunch of layer 4-5 IT neurons from the same data set. Again, here are the somas. These dots are the projections colored by cell types. And you can see that the neurons that project here and projecting here are different cell types. But not everywhere in the brain is well described by cell types. So in a lot of regions, as Carl mentioned, that you have these transcriptomic gradients. So we've been thinking a lot about how to think about this relationship between transcriptomic gradients and projection, which follows topography, which is also a gradient. So here's an example. This is a separate experiment where we're mapping thalamocortical projections. And so each dot here is a neuron in gene expression space, color-coded by the projections, the strongest projections of these neurons. So these two metagenes can explain quite a bit of variance in the projections, which is not surprising. But what's really cool about this is that metagene 1 actually aligns with the topography in projection. So if it's low in metagene 1, then you get these projections to really frontal areas. And if you're high in metagene 1, then you get to these temporal areas on the back of the brain. So metagene 1 actually recapitulate the AP topography of these projections. Now, what I didn't tell you here is that this is actually done in marmoset. So we optimized the BARC not only for mouse, we can do this in marmoset and macaque as well. And this is really interesting here because a lot of these projections are to primate-specific areas. So then we can ask, are these really primate-specific innovations? And what we can do is we can do the same experiment in mouse and find the corresponding gene axons in mouse, and then we map their projections in the thalamus as well. And then we can ask, do we actually see conservation of this kind of relationship? So to summarize, I think with these barcode connectomics techniques, because of the dense labeling and the ability to apply across species, we can now look at gradients, including gene expression axes, and then these projection topography. And we can do comparative studies across mouse, marmoset, and macaque, and using a multimodal approach, combining gene expression and projections. I think, the state of these techniques, I think, is that the technology is not scalable enough to do this kind of studies at a whole brain scale. Definitely in mouse, in marmoset, we can probably do it with some good planning. With macaque, we still need to do more tech development. And there are new technologies in the field, both from our lab and other people in the field, that are continuing to push for higher resolution connectivity. So for example, getting to synaptic connectivity. But I think a critical bottleneck we're experiencing now is the analysis and the conceptual framework. Because I think we're used to thinking about, for example, how cell types project to, whether a cell type projects to a single area, but how do we think about cell type specificity for topography? Now, what if the cell type set is also a gradient? How do we think about this type of relationship? And so I think these are problems that we need to solve as a field in the near future. Thank you.
So let's move on to the next talk. And this one is from Peng Yin from Harvard Medical School.
Yes, so my name is Peng Yin from HMS System Biology and Wyss Institute. So when I joined the panel, I was particularly intrigued by one question posed to the panel, that is, can the current molecular atlasing and effort, and maybe also the connectome effort, be scalable to the entire human brain to that scale, so that we can potentially have a volumetric and brain framework for the graduation of different modalities. So along that line, I'd like to discuss some effort we have been making in this direction, particularly in the volumetric brain mapping using technology called ACE. So I think here is a game that we are talking about the entire human brain, not sparse sampling, but rather the volumetric coverage of the entirety of a human brain volume. We're looking at 180 billion cells, 1.3 liter volume, and potentially, I think the goal would to map tens or even 100 protein or RNA markers, hopefully for more cells or every cell in the brain. So just a sheer volume scale means that it means one thing, that is we can no longer just work with a typical comfortable thin sections, 10, 20 micron tissue sections. We really need to go thick, right? So go to very thick slabs that we can visualize and that will potentially give a pathway forward. So here we're talking about not only 100 micron or 200 micron thick tissues, but at least a millimeter thick tissues, big slabs that potentially can provide a logistic trackable way to scale to the entire human brain volume. If you look at thick tissue imaging, high-plex imaging, and this immediately present a potential scalability bottleneck, since to overcome the so-called spectral limit of fluorescent imaging and the traditional, the natural ways to do sequential cyclic imaging and for the different markers through multiple cycles. But if we apply, I think the traditional iterative imaging approach, for example, for protein, if you use multiple cycles, primary antibody staining, stripping, and imaging, and then for the millimeter thick tissue, each marker, each cycle would take you the switch time about 4 to 5 days, just because antibody and penetration and staining of the markers take time for thick tissues. So if each cycle just switching take you 4 to 5 days, and then for 100 markers, that potential can take you several months to half a year. To address this particular challenge and also other challenges, we developed technology, workflow we call ACE. And the idea is that we bypass a multiple round of staining with the primary probes, but rather we use a barcoded probe. Those are barcoded primary antibody or barcoded fish probes to stain all the targets in one single round. And then after we make all the targets to become barcoded entity, we use an in situ amplification method to turn all the orthogonal barcodes into long concatenators. And this will serve as scaffolds to recruit in a secular fashion and small fluorescent imager strands. Then this through relatively rapid cycles, we can achieve scalable high-plex sequential imaging for millimeter thick tissues. So core to the challenge is how can we develop a high-plex magnification method that is compatible with millimeter thick tissues? Turned out it's not a trivial task, and we initially tried just use a pre-synthesized amplifiers. For example, we developed technology called SABRE, which use a pre-synthesized long concatenator to bind to the DNA primer to provide amplification scaffold. But that works actually for thin section very well, and 100 to 200 micron also work well. But when we can go to millimeter, we really have penetration issue there. So this suggests that in situ amplification would probably be a preferred way to go. But when we look at existing in situ amplification method, HCR and RCA, they have also potential limitations, especially for protein imaging. For one thing, with the high-plex and single run amplification, HCR not only give you 10-plex for the commercially available entity. But more fundamental is that both HCR and RCA, and essentially once you kick off amplification, the amplification will just continue autonomously, which is wonderful for flow perspective. But for thick tissue imaging, this uncontrolled progression amplification could quickly deplete local resources, especially for protein, and this would lead to uneven staining and across the depths of the tissue. So to address this challenge, we developed technology, this is called ACE, and basically is a way to achieve synchronized high-plex uniform extension for the primer. So without going into too much details, basically we have this extender strand which can bind to the primer A, and then at low temperature they can bind, and then polymerase can then extend A to be AA. And then we can increase temperature, at higher temperature, A star, the extender will fall off. And then we cool down again, it will bind again to the end, and then we can extend one more step. So essentially, it's almost like a linear version of a PCR. Through multiple cycles of a thermocycling, we can grow concatenator in a stepwise thermocycle synchronized fashion. And this would provide amplification across target, across tissue depths in a uniform fashion. So I think running almost out of time and very brief on data. So this first validated in thin section, they still, I think, 10 to 20 micron tissue section, but demonstrate a high-plex amplification. 33 protein targets and 30 RNA targets amplified signal. And this is much thicker now, 200 micron and 22-plex protein imaging. And then we go to millimeter section, so shield treated tissue and 10-plex protein imaging. And more recently, we also use another clear method called DISCO, and this is a 16-plex protein marker, again, 1 millimeter mass coronal section. And this is 100 times thicker than the typical thin sections. So yeah, to summarize, the ACE provide a way to achieve scalable millimeter thick tissue imaging, and by using barcoded single substanding with a thick tissue compatible simultaneous circle signal amplification, followed by cyclic exchange of a small image strands. And this new workflow essentially reduces previously 4 to 5 day target switch cycle now to down to, I think, 8 hours in this example here. So now we are looking at, if we talk about 100 markers, and potentially what could have taken half year, now potentially can be condensed to a couple weeks. Of course, to go from here to entirety of human brain, there's still lots of considerations and opportunity and challenges. Happy to discuss more. I think I'll just make another very quick comment regarding the practicum. I think in parallel to the development for the scalable human brain imaging for molecular analyzing, and that's also very exciting development in the connectomics community, in particular using expanded sample and then using light microscope. It appears now people can really visualize the connectome and moving beyond the electron microscope, which is intrinsically less scalable. So if you look at the convergence of the two technology trend and potentially molecular analyzing using this volumetric CT imaging expanding to the entirety of human brain volume, and then in the connectome community, you can also push. For example, there are light-based, light microscope-based approach to the entirety of human brain. And then we are looking at potentially unified approach to unifying the two exciting trend. I think I'll stop here and take questions.
Thanks, Peng. Time for a question or two. Carl.
Yeah, I have a comment. This is great. These methods are really important. I think one thing that you didn't mention, which is really limiting any kind of imaging-based approach to molecular atlas, especially for proteins, is the embarrassing lack of affinity reagents, right? I mean, so if you even think about the synapse, we think there's probably 1,000 different proteins there. We have good antibodies, affinity reagents for maybe 20 or 30 of them for kind of a canonical glutamatergic synapse. So there's a lot of work to do there.
I fully agree. I think for protein imaging and more general for protein detection, and it's not as easy as nucleic acid, where you can just design based on sequence complementarity to design probes. And for protein, the antibody remains to be a key element. Unfortunately, I think in the past 10, 20 years, there has been lots of investment in this direction. For example, NeuroMap and other NIH investment generate lots of open source antibody. And in the coming years, and we hope that this can continue. In parallel, I think that I'm very encouraged by the in the AI community, people seem to achieve lots of progress, if not tremendous success yet, using AI design bundles. And there are maybe molecular ways to turn those bundles into robust, designable affinity reagent for the protein targets in the brain context.
Let's move to the next talk from Dawen Kai from University of Michigan.
So yeah, when I was assigned to the panel, and then I was partially chosen, but also fascinated about still the challenges that we face on the common coordinate framework. I'm sorry that I bring back this topic, but I think Yongsoo and also Carel show very fascinating results from their groups directly work on the common the CCF. But I would really like to use the next few minutes to share some of my thoughts and I might digest of the challenge and more importantly, I think opportunity and maybe some action items that the group, the consortium can do. first, we need to generate a CCF, right? So what CCF is really, to me, is really a boundary definition task. So historically, we really used the anatomy histology based expert annotation to do that, right? Giving the Ellen CCF version 3 as an example here, the multimodal imaging, even though it is not completely all coming from the same brain, but building upon a MRI-based template, everything gets registered, and through the higher resolution image iterations, and the neuroanatomist can use multimodal information to really delineate these boundaries between the subnuclei more clearly. And then after that, a 3D model can be constructed. And obviously, once you have that, the utilization of the CCF will be the next important task, as Carol also pointed out in his research, right? But the accuracy, registration accuracy, I would argue, especially for new data modalities now presented to us, the opportunity of all various different genes, activity, function, and so on and so forth. But how do we do that? So giving one example taken from Xiaowei's group, collaboration with Alan, again, with the MERFISH-based spatial transcriptomics, the registration was done with some of the other contrast staining, for instance, nucleus staining in this case. And then with thin section with 2D registration to 3D. And again, the computational pipeline in this case becomes very important. But of course, the consortium funds a lot more other spatial transcriptomics technology. You also saw that the Barseek, StarMap, and SlideTag, also including some more other commercial platforms, the 10x, etc. The challenge there, I would see as utilization of the CCF is, first of all, is the individual variation. And then associated with that, related to this morning's panel, would be the disease state. So for these two, I may argue, or some of you may also argue, we probably would never get a ground truth. And what is a disease state ground truth? It's probably a continuum. It's the individual variation. When it's large enough, then how do we know that it is healthy versus disease, right? So these are just some of my thoughts I want to throw out here and for later discussion. But on the flip side, I also see some new opportunities, and maybe along with technical challenge and even just philosophically, we need to think about it before we take a move, is that with the new BICAN and Connect data sets, right? So integrating single cell resolution, molecule and morphology information is important, but molecular and morphology neuron subtypes, if we define it in our current canonical way now, they're intermingled. Really, the brain is like the electric circuit, right? The same parts can be reused over and over again in different places. So that is a question: how do we integrate, and to what degree, and what granularity, that how small brain region that we can use that use this type of information to define? And then the other thing in this current pipeline is, whatever way that we were or created a common coordinate framework, whatever imaging modality that we use, it is also we're facing a challenge of averaging a lot of sparsely sampled single cell data from many different brain area, different brain works. So couple that with the individual variation, we might need to really seriously estimate what is the statistical power that is needed before we can draw a conclusion. Maybe that kind of estimation needs to be discussed. And then I would have another thought. Also, Carol pointed out a little bit in his talk about long-range projection and use different color or different Cree lines and molecular identity to really define different region of the subregion of the nuclei. But I would say that, especially with Connect, with the EM data or the expansion light microscopy connection data, whether local connection motif can be used to really define functional boundaries or anatomical boundaries, molecular boundaries together. And then I think if we wanted to really, and Partha also raised this point, if we really wanted to get a really good reference and very good transformation pipelines, I think as a consortium, we might need to include a consensus about what is the reference contrast for each particular modality of data collection moving forward, so that we can make sure the AIs or other language model, learning model can be used to do the stitchings, to do the transformation, to do the registration. Yeah, I'll stop here.
Open for questions. Partha.
Hi, sorry, I kind of said this
Hey,
before, but of course, one could go and collect a reference brain from for every modality. That'll be very hard. What one kind of wants to do, and what I think we do anyway, this is practically what we do, is learn the mapping between the different contrasts. And what we found is that you can do that effectively. And then you need really one reference brain with the richest kind of local texture. For us, that's Nissel. If ideally there was a morphish brain with dense, that would be the ideal reference brain. But then on the other hand, the spacing in the morphish data is somewhat sparse. So I think rather than gathering many reference brains, like one very good, I mean, not one because you have individual variation, but a few high contrast, high textured reference brains, and then learn the other mappings. That's kind of the, I think approach, because you're going to get individual variation, and it's a mess.
Yeah, from your experience, Partha, how transferable these learning models are. Let's say if you learn it from one brain region, can you really use it for the other brain region as well?
No, not from brain region to brain region. You need the whole brain. But I'll give you an example. Xiao Wang sent us some, I don't know, star map, like a little section of a brain, but we could map it quite nicely to the reference atlas because there was DAPI in that little section. And the DAPI was enough, there's local information. This is really related to what people call foundation models nowadays, because you're kind of learning a local embedding. So what does it mean to be at a point in the brain? It is defined by the context of that point. What kind of cells are around you? If you look at Nissels, you're getting a kind of poor person's, estimate of that cellular neighborhood. If individual variation is very small, then you can just rely on a cross-brain mapping using just coordinates. But otherwise, one should weight these local textures. So you kind of want the reference brains with those local textures.
Next is, go ahead. Yeah, David and Yongsoo.
Thank you for the really interesting talk. So I was struck by your terminology, disease state CCF. So I just want to clarify a little. Are you referring to like this morphological deviation by particular genetic model or environmental insult, and to kind of define, like if you have this, the Fragile X or a certain gene, and is that what you mean by?
To a certain degree, it's almost like a chicken and egg question, right? If you have a developmental disease, you distorted the morphology, and how do you define where the brain region that is? So that's why I meant disease state when I address that question.
Let's do one more from David.
Yeah, I just wanted to follow up on a small technical point on what Partha said. Partha's lab actually produced 12 very beautiful Nissel brains that we used in a paper to automatically look for texture as a project with Yoav Freund. So those are available, the entire sets sliced at 20 microns. And the sort of animal to animal variability, which we believe is at the biological variability limit, is about 90 to 100 microns, I think similar to the numbers Karel spoke about. So those are available.
Thanks, Dawen. Let's move to the next talk. This one is from Nuna da Costa from Allen Institute for Brain Science.
Thank you, everyone, for inviting me to this discussion. I bring the perspective of starting this journey of multimodal interaction from this high resolution EM data sets, and that are at the scale of millimeters for now. And one thing that we've seen a lot that has surprised us is how remarkable the specificity between cells are, the specificity of connections between cells and cell types in these data sets, between excitatory cells, between inhibitory cells. You just pick a cell type and you see it. And then, of course, we're interested in related this to other modalities, because we want to know how does this specificity of connections is reflected in the activity of those particular cell types, how this specificity of connections is reflected in the information in the genome that was used to construct or to self-construct this specificity of connections. And we have recently published several papers showing this specificity, but for the topic of discussing challenges, I'm gonna use an example that is very fresh. It is incompleted. Is it unresolved? The cross modality matching, but I think it's very interesting and aligns well with the conversation that started with Karel, because actually Jayaram, Chandrashekar, that Karel already mentioned, that came to us and asked, can you find in your millimeter scale EM data sets these accents for which I have this beautiful all mouse brain resolution, and they are, we think, modulatory accents. So I went hunting in the EM data, and I hunted for accents that have hallmarks, ultrastructure hallmarks of this ultrastructure hallmarks of that are not just canonical phenaptic, and we find several of some. That the one is represented here on the top. It's an accent that goes through layer 1 and then falls down these grapes of boutons that form synapses, and most of these synapses, these axons making just one synapse with a postsynaptic target, but there are a lucky few, 2 or 3 per axon, that goes from one synapse per cell to 70 synapses, 40 synapses. It goes to town on these synapses. It really wants to, it goes to town on these cells. It really wants to connect to these 2 or 3 cells. And these cells, which you see here on the bottom right, they have in white, and the colors are this kind of hyper-targeting axons, and these cells have morphological and connectivity properties that suggests they are from the same cell type. So and not only that, but when you ask these cells, is there any other axon that really is obsessed with you, there's another 1 or 2 additional axons that you can see here in multiple colors that not only hyper-targets that cells, but hypertargets the same other 2 or 3 cells that the first axon hypertargets. So remarkable specificity of cell type connectivity, remarkable specificity of single cell connectivity. And the question immediately arises, what is this? What is the cell type of this presynaptic axon? Where does it come from? And what is the cell type of the postsynaptic axon, or even the subclass? And in the past, and currently, when we need to try to match with other modalities beyond morphology and connectivity, we have taken advantage of the fact that it's this beautiful data sets of patch-seq data, of which there is morphology, and here the patch-seq is on the pink side and the EM is on the blue side. There is morphology on the patch-seq, there is transcriptomics on the patch-seq, there is electrophysiology on the patch-seq, and on the M side, on the blue side, we have morphology, and then we have the connectivity and other subcellular features. So we can use kind of morphology as a kind of ancient Greek in a Rosetta Stone of modalities to link between the two, and this is something that we've published recently, the work of Claire Gamlin, and it's a collaboration between Stacy Sorensen, Forrest Coleman, and myself, and in some cases this works very well. So you see here some cases where 2 cases of which this paper focus on, where there were all SST cell types, and on one side you see the morphology from the patch-seq, the other one the morphology from the EM, and there's another secondary here on the right, again the morphology from the patch-seq from the EM, and this one on the right turns out that this SST, Martinotti cells really like to target ET neurons, layer 5 ET neurons, and the ones on the left really like to target layer 5 IT cells. But this discussion is about challenges, and one challenge that exists in doing this is that we really don't have ground truth. So we don't have a data set that has both PET-seq and EM data with which the neurons are properly paired. A second best will be a data set of which at least there are some anchor cells for which we have EM and PET-seq data. We also don't have that. So we are bound with data sets of which there is no overlap, and we have to do feature alignment to be able to match the cells, because even though it's morphology, they were collected in different experimental conditions, and so that's one of the technical challenges. I'm not going to go into details in the resolution, but the nice thing is that at least there are some cell types that even though the cells in themselves in the data sets cannot be paired, there are cell types that everyone agrees, no matter if you come from transcriptomics or morphology or connectivity, they're the same cell. There are not many, but there are a few, like, for example, the Shenlia cells. And cells like that can be used as anchors. And on top of it, we live in a world that it's kind of we're lucky to live in a state of the technology that we now have not only genetic tools that can be targeted to individual cell types that can serve as confirmations of the findings of the feature alignment, and was already mentioned expansion microscopy, with liking that further you can go and then check if your hypothesis that comes from the feature alignment is correct. And so, for example, for this particular target cells that I mentioned, they seem to be the CCK positive, VIP positive basket cells that are hyper-targeted by these axons. There is another challenge that I want to bring in, and Karel already touched a bit on this, is this cross-modality diversity. And I pick here one example from the PET-seq and transcriptomic cell types, just because I think it's better for this audience, but there are, if I start from connectivity, I'll have equally examples of diversity. So here's a T-type that when you look at the morphology and even the electrophysiology, really has two very different groups. And if you ask an anatomy what this is, they'll say it's a Martinotti SST type, checks with the T-type, but this one, any anatomist that will look at it will tell you that it's the kind of classic anatomy of a basket cell, and actually the electrophysiology properties also match with parvalbumin cells. So we need, and I don't think this is a challenge of the technology. I think this is a feature of nature, a feature of biology, that within, no matter where you come, which modality you come from, there is really a diversity on the other modalities, and it's, instead of thinking on a model and being dogmatic that it's going to be a cell type to rule them all, we really have to work towards making sure that there are the passes to integrate across these different modalities and explore these clusters. There seem to be in one modality actually have a diversity of shapes, of function, of connectivity in the others. And with that, I want to acknowledge all the people that contribute to this work, and of course the funding, including the Brain Connects.
Right, thanks, Nuno. Let's take maybe one question, if there is one, before moving to the next talk. Vatsala.
I was just wondering, you said there's some cell bodies where a lot of these neurons are, making connections to. And is there just something specific about this cell type, special about this cell type, that so many neurons want to connect to it?
Yeah, so our current estimation of the target cell type, and it's not just a cell body, the exon starts on the tip of a dendrite and then climbs through its synapse, goes to the soma, synapse, lives on the dendrite, synapse. It's really hyper targets them. And these cells are usually they are very spiny, which is not typical of basket cells and inhibitory cells, and they target the somas, and they have lots of morphological hallmarks of the CCK positive, VIP positive cell types. So they don't hyper-target other basket cells that are in the vicinity, or somatotaxin cells. When they get obsessive with this particularly cell type.
And do you see any like difference in the single cell transcriptome of these cells? Like, are they morphologically different than the other?
When we look, since we don't have gene expression for the EM data, but when we use the PET-seq to match it to the gene expression data, yes, they have morphologies that are similar to what we see. That's how we are able to do the connection, and then we can actually ask, are there particular receptors that are expressed on this type that start to give us hints of what's that incoming axon, and that's that and other data is even though we have not absolute proof, makes suggestions what's that incoming axon, which is where the conversation with Jayaram started.
All right, fantastic. Let's move to the next talk, and this is starting theme 2, which is the conceptual challenges in using these data for discovery and hypothesis generation. So the first one here is from David Kleinfeld from UC San Diego.
I have an idea. There's a problem here with this machine. Why don't you let one other person go before me, and I'll try to straighten it out, okay?
Sounds good. So then we'll switch. Next one then will be Burke Rosen from WashU, if you're ready, Burke, and then we can go back to David.
My name is Burke. I'm a postdoc in the Van Esten and Glasser lab at WashU, and my background is in neuroimaging, but I've been moving towards bioinformatics as part of our collaboration as part of the HMBA, particularly with Helen and also with some other folks. And that's been really cool. And so I'm going to talk about a couple of practical steps about how we can bridge these disciplines. All right. So neuroimaging and bioinformatics have a lot in common. But they've developed mostly independently over the past 3 decades or so. So they both involve very large, high-dimensional data sets, and those are stored in large, complex formats. They have foundational multi-institutional projects like the Human Genome Project or the Human Connectome Project. And because there's a lot of smart people working in both of these fields, there are deep reservoirs of analytical tools and expertise that we can leverage. And my pitch for this talk is that we can tap those reservoirs and accelerate new discoveries by packaging our data into structures that the other field can more easily understand and interpret. And the main example I'm going to go through is going from bioinformatics to neuroimaging, because that's what I've been working on recently. So spatial is super important for this because it adds space to bioinformatics, and that allows that space to be a common substrate between the transcription data and neuroimaging like MRI. And so I've got 2 data sets here. On the left, I have a pretty standard spatial image. This is macaque neocortex. And in it, of course, every cell is a dot, this discrete spatial entity, and each dot is labeled by its type from expression or T-type. And this looks quite a bit different from MRI. This is actually from the same animal. We have functional MRI from a retinotopic task and structural MRI, architectural MRI. And this data has been projected onto the cortical surface because we're looking at neocortex, and then flattened so we can see across all the folds. And we've zoomed in here to the V1, V2 boundary. And that's actually the same region that's being shown in this spatial slide. Now, as a neuroimager, when you show me a bunch of dots in this 2D plane, I don't really know how to handle it, and neither does our software, neither do our tools. So the first step I had to do to really get a handle on these data is to convert them into continuous cell type density maps. And this is a fairly easy step, which I think goes a long way to bridging these disciplines. And that is, I essentially drew voxels on the slide and then counted the number of cells of each type in each voxel. And then for each type, I have a normalized map of that cell type's proportional density. And already we can see with this very simple transformation, the data has become a lot more similar to MRI. And if we were in subcortex, we'd be able to do the same sorts of volumetric analyses on it that we do to volumetric MRI. Now, because this is neocortex, we did several additional steps because we want to get it onto the surface so we can properly analyze the variation across the convolutions. And to do that, we had to figure out precisely where these slides are This is the registration problem that many speakers have addressed today. And that is, of course, quite difficult. But once we've figured out where those slides are, we can project it to the surface, see where our spatial data is, and then interpolate between our samplings, our sparse samplings. That is quite a bit more difficult than simply converting things into continuous fields, but it's beyond the scope of what I want to talk about today. But having done all of that, we can then take the spatial data and project it to the surface in a format that looks a lot like this MRI. So in this middle panel here, this is the same transcriptomic type. Its density projected onto the cortical surface. And now we can make direct comparisons between MRI and spatial. And we can see that the boundary between V1 and V2 comes out very clearly here because this is a V1 enriched cell type. That was always baked into this slice. We can see a V1 here with the higher concentration of the cell type and then V2 with the lower concentration. But now we can do this direct comparison. And what I think is really cool and even more exciting is now we can take tools that were developed for MRI and apply them to the spatial data. So this is a gradient map. And in MRI, we produce these gradients or spatial derivatives with respect to the surface in order to detect areal boundaries, because the gradient is going to be high. It's going to form this ridge where the properties across the surface are changing rapidly. And so now we can apply the same technique easily because the numbers have been put in the same format to the spatial data, and we can see this prominent gradient ridge right across the V1, V2 boundary. And so that's really cool. To give an example going into the other direction, and this is more prospective because I haven't actually done this yet, it's just an idea at this stage, but UMAP and T-SNE decomposition, which we've seen again several times today, is rare in neuroimaging, but I think it has great potential. And I'm sure there's all kinds of other tools that have great potential that just haven't been applied to neuroimaging data yet. But a cell by gene expression matrix is really not that different from a voxel by time point fMRI matrix or vertex by time point fMRI matrix. They both have spatial relationships baked into them and meaningful correlational structures across either time or genes. And so if we could repackage fMRI into H5AD files and ADA files, then this whole universe of tools is unlocked and expertise. I haven't done that yet, but I'm excited to get a chance to do so. And again, my take-home message is if we can just repackage our data in, I think, fairly simple ways, then we can really accelerate discovery by borrowing each other's tools. Thank you.
Right, great. Thanks, Burke. Question from Mike.
Yeah, Burke, that's really interesting, and I know that we've taken advantage of some of the approaches that you've shown for the flagship paper for the BICAN, which is exciting. What do you think is I mean, the last item that you mentioned about potentially using these kind of latent space representations, what would have stopped anybody in neuroimaging? It seems like such a basic idea. I'm kind of surprised that hasn't been picked up.
There are latent space representations in neuroimaging. I mean, the simplest, going back a very long time, is principal component analysis.
ICE too. I mean, ICA, right?
Yeah, ICA, PCA. But I think there's perhaps because the dimensionality is so much higher in the gene space. I think there's been greater development, at least in the past couple of years, in the bioinformatics space than there has in neuroimaging. In that particular subregion, whereas a time series analysis has a ton of progress in neuroimaging, and if those sort of autoregressive techniques could be applied to gene series, then that is potentially useful. But these communities don't talk to each other that often. And the existing neuroimaging and bioinformatics a crossover has been mostly more in sort of a GWAS sort of space. But now with spatial, we're really talking about the same things. We're talking about the same structures at the same approximate resolutions. I just think there's a great potential for cross-pollination.
So I think David is trying to re-enter the meeting. It looks like he needs unmuting access. I don't know if that's something that the administrators can see.
Okay, great. So let me thank Xiaowei and Jeremiah for this opportunity. So we're coming into this as a research group and part of two U19 brain circuit programs that are very much interested in circuits. And what I want to propose here is this very foundational need for BICAN to consider adopting standards to populate an annotated graph, a way to actually use all this data, connectivity data, cell type data, and then make circuits that can make predictions about neural dynamics and about behavior. And this is clearly at the EM level, and I'll discuss briefly how, and it was already raised by Nuno, how this could be downscaled. So it will, you can use it to estimate mesoscopic connectivity, which is where most of the data is. And you could also go in the other direction. Again, Nuno raised this point, taking mesoscopic data and within statistics, populating an EM level graph. Okay, next, please. So we've seen, increasingly sophisticated connection matrices. This just happens to be a particular example from my colleague Hongwei Dong, where you've measured connections between two regions, both in the anterograde and retrograde direction. Even at the mesoscopic level, you find you get regions of the brain with particular functions, something already identified by Larry Swanson. Sensation at a distance, sensation at close becomes clear from the way the connectivity matrix factors, and most impressively about mesoscopic measurements is the possibility of doing two or more electrode penetrations and measuring pairwise connectivity and actually getting PSPs. But from the point of view of making circuits, this is really kind of leaves you at the sort of mean field approximation level. It's not going to be precise. So next, please. Okay, so of course we have connectomic.em connectomics now. This is an example from Maurice Helmstadter, but these are very restricted. But I think the point is that now is the time to start to formulate data in a graph, annotated graph, which is basically a matrix. So just with reference to a little bit of maybe even seemingly naive mathematics on the bottom, you really have a matrix, which is given by the square, of the number of neurons in the brain. Of course, the number of non-zero elements is relatively small. And this takes you all the way from sensory inputs, both external inputs, exoreception, internal inputs, interior reception, through sets of synapses, and then has feedback between all of the neurons, and then takes you over to the output of motor neurons. And at the end of the day, this is the kind of representation we're going to need if we're going to be able to make a connection from neural dynamics and the design of circuits to actual behavior. And in fact, of course, since the neurons or the motor neurons are actually driving the muscles, and the muscles are what are driving the distortion of our body sheet, I mean, this very much ties into behavior. And I mean, there's a number of sort of considerations that is going to make it imperfect. Annotation will consider all the things that were raised here at this BICAN meeting, transcriptome, lineage, etc. but there are certain approximations that are made. From EM, you don't measure PSPs, at least it's not obvious. It's dead tissue, but you do measure number of contacts, which is not necessarily the same as PSPs. Okay, next, please. So the one thing that comes up is how do you go from a vector of all the neurons down to mesoscopic groups? I mean, this is some manner of coarse graining, very familiar within physics and chemistry, using overlap of known descriptors, transcriptome, shape, etc. On the other hand, how do you fill up this EM level? Connectome is at least starting now, and the bulk of the data by far is mesoscopic data. And here within sort of the distributions of known properties of mesoscopically similar cells, when one just wants to expand on that representation. And there are some things, in fact, that you may never know through EM, like I mentioned, PSPs or epigenetics or proteomics, etc. Okay, next, please. So there's obviously many challenges. One is that at the sort of mesoscopic level, we usually think of things in a very deterministic manner. And as we move to the cell by cell level, I think we have to understand, we have to accept a probabilistic specification as we move from to a cell-based, connectomic scale. In some sense, this represents animal to animal variability. But I think the field itself has to be more understanding of probabilistics. We really need to try to get also down to the level of biological uncertainty. We're not quite there yet. We also need, quite to understand in many things like transcriptomics, at what point these dendritic trees, these dendrograms are sort of going into the noise. The biggest thing in terms of circuits is the mapping between synaptic contacts and postsynaptic potentials. Okay, one could go on, but again, I think the major thing is to think in terms of these high dimensional graphs. So last slide, please. So, I mean, is it feasible? I mean, there's a huge number of entries. If you think of the mouse, there's potentially 10 to the 15 terms. When I mentioned, most of the matrix is filled with zeros. So there's a smaller number of entries. It's about 10 to the 11. It's really not crazy to think about this, and certainly at much smaller scales, this has been attempted, and just because of other work at the sort of level of matrix with about 10 to the 7 entries, which is the brain vasculature, you could already do calculations on these kind of graphs. So I would just put forward that as a community, BICAN should really consider how to set up a graph in the best way and what the standard would be, and how to ingest data. And I think what we really want is, if you read a lot of theories papers today, they basically turn cortex into a front-loading washing machine, as shown here in the little cartoon on the bottom. And this is not the way it works. And we really want everybody to start thinking about the importance of this detailed information, if in fact it holds critical boundary conditions on computations in the brain. Okay, so thanks.
Jeremiah and Xiaowei, I just want to remind the time. Now it's about 3:50. And so I would suggest maybe let's end this panel discussion in 10 minutes so we can have the last plenary session at 4 o'clock. Okay?
Yeah, sounds good. Well, we had a lot of questions at the end of each talk and discussions and so on. So 10 minutes sounds great. Let's help keep everything in the time limit. So yeah, so we're open to general discussions. But this is a session primarily about how do we integrate multiple modality of measurement, and then how do we extract and synthesize the knowledge from integration of a different kind of a modality. Most, of course, experimental discussions and computational discussions are very important. So we'll see who wants to, there's a hand raised, right? Already, I can see that. Zhuang.
From end user point of view, I definitely feel very excited about this whole session discussing the technical and the conceptual tools. I just want to quickly raise one question. Consider the CCF that Carol, Yongsoo, and Dawen has mentioned. With one of my own user experience, when we try to apply the CCF to actually for the disease context. When we have, say, like a mouse brain that deviate quite a lot from the normal variation, say a mouse brain that carry neurodegenerative mutation, hippocampal is largely degenerated, lateral ventricle become a large hole. The beautiful whitehead template or whitehead brain atlas doesn't really register with the standard tool, although we haven't tried the new ones like the one that Pathon mentioned. I just want to ask the expertise here for this type of sort of like the future disease application will be the best consideration or practice to move forward.
Maybe I can chime in a little bit. To how you ask a very tricky question, I think, we kind of encounter similar problems with some collaborations. I feel that there needs to be some sort of like molecular markers to set up where the healthy or normal part is of the brain, or nucleus and sustain like Patha indicated, and maybe use that to set kind of scale, overall shape, and then leave whatever the places that's deviate from the standard, and that's what it is. If the whole brain is so messed up, the whole thing gets just like really distorted, then I guess it's hard. But it's always need to be think ahead of time before each experiment. My experience was, what is my reference point and what is my standard? If condition permits, I also found that sometimes the Amazon, a lot of times we do these 2D section samples. There's also another big challenge is that registrating the 2D sections back to the 3D references map. Not only just how it registers, but also the orientation and all that. So sometimes taking a 3D scanner from Amazon, do it before you do the sectioning with what the brain looked like and all that would be helpful. So be creative along each pipeline step, have references.
Yeah, so I do want to say that given we have limited time, I want to bring up a couple of things. One is while mouse CCF registration, the individual measurement to reference maps and so on is certainly still have challenges. But the challenge for CCF registration for the bigger brains will certainly be much more, whether it is a non-human primate or even human is, even more difficult. So if there are discussions along that front, that would be great. And then the second is that this is a session, we have CCF is always an important thing, and we have also discussed that on the first day. And this is also a session about integration. About integration of different modalities, molecular and morphological connectivity and functional. So it'd be great to also devote some time to this area of discussion.
So it looks like in order, at least Nuno, Yongsoo, Xiaoyin, Karel.
My comment will be brief. I think just to say that David's vision is a huge opportunity of integration between Brain Connect and BICAN, and not just for BICAN. And even on the Brain Connect side, there's already this vision that aligns with what David said, of having this common connectivity matrix, which is an annotated matrix across the whole mouse brain, aligned with the CCF. So yeah.
Yeah, I want to chime in. I think what David proposed was brilliant. But I want to ask David a question. So there is this Weying diagram that has particular spatial coordinate one to the other, and there are vascular coordinate also has geometry, but there are also like molecular signatures that may or may not have a spatial data as well. How you envision to vectorize all of this into the kind of coherent matrix?
Okay, that's I have a partial answer. Thanks for the question. So in a perfect world, if you really did have all the neurons, at that point you could forget about localization, the kind of location information said that Karel raised during his talk, because you know who you're connected to. And of course, implicit in this annotation is something about the electrotonics in the cell. You know how you'd have to know how you're doing synaptic integration, because it takes place over some spatial scale. So, but I think in practice, if I'm answering the question fully, you do have to keep track of some of this location information, because there are some things you just by the definition that you're using fixed tissue, it's not clear you can get a complete transcriptome, or clearly you can get PSPs. So you'd like to be able to always access other data sets. So I think we're at least, in a future which is at the limits of my imagination, you're always going to have to tag the annotation with the location within the brain. And that means pretty good registration, really, at this sort of biological level, which is probably no worse than 100 microns, maybe less.
There are some questions for Carol too. So let's jump the apparent queue and have Carol to answer or talk first.
Okay, well, very, very quick comment. I mean, I think that David's vision is, I do think that's really what connects us about. And I think all of us should be thinking about it, right? Because cell types is sort of the genetically defined cells are kind of the nodes of some kind of an average circuit diagram, right? On which kind of the fine scale specificity that Nuno talked about sits. And I think it sometimes seems kind of silly, but the even very weak anatomical constraints can be incredibly useful for modeling. So for example, that thalamus and cortex are connected through kind of a bottleneck, loosely speaking. Lots of connections, right, within the cortex, but the connections to the thalamus is through relatively few fibers and back. The theorists have made some very interesting predictions based on that assumption alone, right? So I think even partial data, if it's quantitative, is incredibly useful. What I wonder is, do we actually have the tools to? Are there some kind of software tools, databases that deal with all the spatial and discrete variation that ultimately require building this kind of a circuit diagram? Or do we need a kind of an infrastructure that is missing? I'm not aware of something that I would right away jump on to build such a circuit diagram. This might be worth discussing.
We're almost out of time. 2 more hands are up. Please be very brief with your comments, Xiaoyin and Partha.
So I wanted to comment on the part about registering like non-human primates. I think we've had I have some intuition on this just based on our experience, sometimes even registering to the wrong species, like Nemostrina registering to rhesus. I wonder if people have ideas about using the overall transcriptomic identity in spatial data to do this kind of registration. So not using matching markers, but actually just using borders that you can see in spatial transcriptomic data. That seems to be very powerful and very accurate in those cases.
Yeah, I agree. So Partha, can you very brief? Yon is eager to start the next one.
Two quick points. One is Karel tools. The first tool is language. We break the brain up into parts. We label different things, and we even talk about the relationships in terms of circuits. You read the literature, you're relying on language. And one tool that we are not talking about is various natural language processing. Now there's language models. I think, logical reasoning, all kinds of stuff. There's software around this, and that can be matched up to the literature and the data. So that's one set of tools. The other quick point is that we love talking about regions and mapping. but one should think about the circuits and the systems as well. And that's a different way of organizing the data. Even in nomina anatomica, you have your head, which is the region, but then inside it you have vascular system, which doesn't all live inside the head. So just because it's brief, I will stop there, but thanks.
All right, Jeremiah, any final words?
Thanks to all the speakers.
Thank you.
Wish we had more time.
Thank you for all your contributions to this section, and thank you, Jeremiah. And Leon.
Yeah, thank you, Xiaowei, and also Jeremiah for this wonderful panel discussion.